In this video, we are going to tell you about the biological phenomenon which is related to the quantum physics. One is the gene mutation, the other is photosynthesis. Let's look at the gene mutation first. In the cell, there is DNA, RNA, which has the information, and both of them are made up of 4 different nucleotides, ATGC and UCAG. Naturally, they bond each other, A and T, G and C. These bonds are the hydrogen bond. By the quantum effect, the hydrogen can move to the other side. Then the structure of the DNA is changed like this one. We call this the imino or enol tautomer. Interestingly, the tautomer form can bind with the other nucleotide. For example, C can bind with T, A can bind with G, and vice versa. We call this phenomenon mispairing. And this kind of mispairing can cause the mutation. How can mutation occur? The mutation occurs while the DNA is replicating. At the first replication, an anomalous CA base pair formed, and at the second replication, transition mutation occurred. Then how often this kind of thing will happen? We can calculate this by the quantum mechanics. There is a paper which used double well potential model to calculate this. I will introduce this paper briefly. In this paper, they set the hydrogen environment by the double well potential. Then we can calculate the quantum functions of the hydrogen. You can see like this one. Since the tautomer case has a higher potential, there is no possibility for the N=5 case. So we can say that the N=6 case have the possibility for the tautomer form. Plus, the hydrogen bond shouldn't break with this phenomenon happen. We have only certain energy function because of these two conditions. In this paper, they said that N=6 case to N=8 case are available to their assumption. In results, interestingly, the possibility that this kind of mechanism can appear isn't that low that we can ignore the possibility that the gene can mutate by the quantum tunneling. In conclusion, the gene mutation can occur by the quantum effect. Next, we are going to talk about the relation between the photosynthesis and the quantum physics.
From now on, I'm going to talk about how quantum mechanics is applied when photosynthesis occurs. Quantum mechanics and photosynthesis, what does Schrödinger equation have to do with chlorophylls at ATPs or Kelvin cycles? We'll get to that later. So this is the figure that briefly explains the photosynthesis. During light reaction, electrons in chlorophyll are converted into excited states, and in dark reaction, electrons are transported to the reaction centers, produces ATP, and ATP is used energy source inside the plants. In the light reaction, scientists experimentally speculated that quantum tunneling and quantum coherence has occurred. This is ephemeral complex in green surfer bacteria, which is responsible for transferring energy from the antennas to a reaction site. Light excites electrons in an antenna. The excitation then transfers to various proteins in the ephemeral complex to the reaction center to further photosynthesis. In this process, scientists revealed that an efficiency between the absorption of electrons and transfer to the reaction site is above 99%. This high efficiency cannot be explained by classical mechanics such as diffusion models. So what happened? As per classical models, it can take one of the many passes to the reaction center for being absorbed, but that would lead to discrepancy about the efficiency of the absorption of the electrons. So scientists assume that these excisions exhibit quantum coherence, a phenomenon where both waves occupy the same phase and hence interfere positively. Excision can sample multiple pathways and determine the most efficient one, and this would make sense above all. To look at the concepts in more detail, I took a look at the paper published in 2014 in Nature Physics and explained the main concepts briefly. It focuses on the PS2 reaction center. It is in the PS2 reaction center that the excitation energy is converted into transmembrane electrochemical potential with near unit efficiency. On the excitation, the energy is delocalized among these cofactors, and it leads to collective excited states mixed with charge transfer states. This mixing leads to excisions with city character. It is called excision city, which means the excision exhibit a non-uniform electron density, like the figure, and it is excision city that provides ultra-fast channels. Another phenomenon that I want to explain is quantum tunneling when observing light. This pigment shown in the picture complements the absorption of light by chlorophyll. You can see that light energy absorbed by antenna pigments molecules, and it is finally transferred to a chlorophyll, a reaction center, to drive photosynthesis. This is where the quantum tunneling happens. The electron jump from one pigment to another while making its way. So think of each pigment as a potential wall. As we learned in the class, an electron in a finite potential world can travel into classically forbidden high potential regions. Um, so if you want to calculate the distance and the time for each pigment to jump, you can use the calculations shown in the slide. To sum it up, we discussed how quantum mechanics is applied in DNA gene mutation and photosynthesis. Thank you for watching the video.